Biological systems are much more than the sum of their parts. The whole is greater than the sum of its parts. That familiar adage tours the important concept that a combination of components can form a more complex organization called a system. Examples of biological systems are cells, organisms, and ecosystems. To understand how such systems work, it is not enough to have a parts list, even a complete one. The future of biology is in understanding the behavior of whole integrated systems. The emergent properties of systems. Take another look at the levels of life. With each step upward in this hierarchy of biological order, novel properties emerge that are not present at the level just below. These emergent properties are due to the arrangement and interactions of parts as complexity increases. For example, a test tube mixture of chlorophyll and all the other molecules found in a chloroplast cannot perform photosynthesis. The process of photosynthesis emerges from the very specific way in which the chlorophyll and other molecules are arranged in an intact chloroplast. To take another example, if a serious head injury disrupts the intricate architecture of a human brain, the mind may cease to function properly even though all of the brain parts are still present. Our thoughts and memories are emergent properties of a complex network of nerve cells. At an even higher level of biological organization, at the ecosystem level, the recycling of nutrients, such as carbon, depends on a network of diverse organisms interacting with each other and with the soil and air. Emergent properties are neither supernatural nor unique to life. We can see the importance of arrangement in the distinction between a box of bicycle parts and a working bicycle. And while graphite and diamonds are both pure carbon, they have very different properties based on how their carbon atoms are arranged. But compared to such non-living examples, the emergent properties of life are particularly challenging to study because of the unrivaled complexity of biological systems, the power and limitations of reductionism. Because the properties of life emerge from complex organization, scientists seeking to understand biological systems confront a dilemma. On the one hand, we cannot fully explain a higher level of order by breaking it down into its parts. A dissected animal no longer functions. A cell reduced to its chemical ingredients is no longer a cell. Disrupting a living system interferes with the meaningful understanding of its processes. On the other hand, something as complex as an organism or a cell cannot be analyzed without taking it apart. Reductionism, reducing complex systems to simpler components that are more manageable to study, is a powerful strategy in biology. For example, by studying the molecular structure of DNA that had been extracted from cells, James Watson and Francis Crick inferred in 1953 how this molecule could serve as the chemical basis of inheritance. In 2001, almost half a century after the famous work of Watson and Crick, an international team of scientists published a rough draft of the sequence of 3 billion chemical letters in a human genome. Researchers have also sequenced the genomes of many other species. The press and world leaders acclaimed the Human Genome Project as one of the greatest scientific achievements ever. But unlike past cultural zeniths, such as the moonwalk of Apollo astronauts, the sequencing of the human genome is more a commencement than a climax. As the quest continues, scientists are learning the functions of thousands of genes and their protein products. And research is now moving on to how the activities of these myriad molecules are coordinated in the development and maintenance of cells and whole organisms. At the cutting edge of this research is the approach called systems biology. Systems biology. Biology is turning in an exciting new direction as many researchers begin to complement reductionism with new strategies for understanding the emergent properties of life. How all the parts of biological systems, such as cells, are functionally integrated. This changing perspective is analogous to moving from ground level on a certain street corner to an aerial view above a city, where you can now see how variables such as time of day, construction projects, accidents, and traffic signal malfunctions affect traffic dynamics throughout the city. The ultimate goal of systems biology is to model the dynamic behavior of whole biological systems. Accurate models will enable biologists to predict how a change in one or more variables will impact other components and the whole system. How, for example, will a slight increase in a muscle cell's calcium concentration affect the activities of the dozens of proteins that regulate muscle contraction? How will a drug that lowers blood pressure affect the function of organs throughout the human body and possibly cause harmful side effects? Systems biology is relevant to the study of life at all levels. Scientists investigating ecosystems pioneered the systems approach in the 1960s with elaborate models diagramming the network of interactions between species and non-living components in salt marshes and other ecosystems. Even earlier, biologists studying the physiology, functioning of humans and other organisms were integrating data on how multiple organs coordinate processes such as the regulation of sugar concentration in the blood. Systems biology is now taking hold in the study of life at the cellular and molecular levels, driven partly by the deluge of data from the sequencing of genomes and the growing catalog of known protein functions. In 2003, for example, a large research team published a network of protein interactions within the cell of a fruit fly, a popular research organism. The model is based on an extensive database of thousands of known proteins and their known interactions with other proteins. For example, protein A may bind to and alter the activities of proteins B, C, and D, which then go on to interact with still other proteins. The basics of the system's strategy are straightforward enough. First, it is necessary to inventory as many parts as possible, such as all the known genes and proteins in a cell, reductionism. Then it is necessary to investigate how each part behaves in relation to others in the working system, all the protein-protein interactions, in the case of our fly cell example. Though the basic idea of systems biology is simple, the practice is not, as you would expect from the complexity of biological systems. It has taken 3 key research developments to bring systems biology within reach. High-throughput technology. Systems biology depends on methods that can analyze biological materials very rapidly and produce enormous volumes of data. Such mega data collection methods are said to be high-throughput. Bioinformatics. The huge databases that result from high-throughput methods would be chaotic without the computing power, software, and mathematical models to process and integrate all this biological information. The new field of bioinformatics is extracting useful biological information from the enormous, ever-expanding datasets, such as DNA sequences and lists of protein interactions. Interdisciplinary research teams. In 2003, Harvard Medical School formed a Department of Systems Biology, its first new department in 2 decades. Nearby MIT is busy organizing over 80 faculty members from many departments into a new program for computational and systems biology. These and other systems biology startups are melting pots of diverse specialists, including engineers, medical scientists, chemists, physicists, mathematicians, computer scientists, and biologists from a variety of fields. A number of prominent scientists are promoting systems biology with missionary zeal, but so far the excitement exceeds the achievements. However, as systems biology gathers momentum, it is certain to have a growing impact on the questions biologists ask and the research they design. After all, scientists aspired to reach beyond reductionism, to grasp how whole biological systems work long before new technology made modern systems biology possible. In fact, decades ago, biologists had already identified some of the key mechanisms that regulate the behavior of complex systems such as cells, organisms, and ecosystems. Feedback regulation in biological systems. A kind of supply and demand economy applies to some of the dynamics of biological systems. For example, when your muscle cells require more energy during exercise, they increase their consumption of the sugar molecules that provide fuel. In contrast, when you rest, a different set of chemical reactions converts surplus sugar to substances that store the fuel. Like most of the cell's chemical processes, those that decompose or store sugar are accelerated or catalyzed by the specialized proteins called enzymes. Each type of enzyme catalyzes a specific chemical reaction. In many cases, these reactions are linked into chemical pathways, each step with its own enzyme. How does the cell coordinate its various chemical pathways? In our specific example of sugar management, how does the cell match fuel supply to demand by regulating its opposing pathways of sugar consumption and storage? The key is the ability of many biological processes to self-regulate by a mechanism called feedback. In feedback regulation, the output or product of a process regulates that very process. In life, the most common form of regulation is negative feedback, in which accumulation of an end product of a process slows that process. For example, the cell's breakdown of sugar generates chemical energy in the form of a substance called ATP. An excess accumulation of ATP feeds back and inhibits an enzyme near the beginning of the pathway. Though less common than negative feedback, there are also many biological processes regulated by positive feedback, in which an end product speeds up its production. The clotting of your blood in response to injury is an example. When a blood vessel is damaged, structures in the blood called platelets begin to aggregate at the site. Positive feedback occurs as chemicals released by the platelets attract more platelets. The platelet pile then initiates a complex process that seals the wound with a clot. Feedback is a regulatory motif common to life at all levels, from the molecular level to the biosphere. Such regulation is an example of the integration that makes living systems much greater than the sum of their parts. 1. Emergent properties and sentence construction. The relationship between a sentence and the alphabet of letters from which it is constructed is a classic example of emergent properties. The individual letters of the alphabet have no inherent meaning on their own, but when combined in a specific way, they form words, phrases, and sentences that convey complex ideas and meanings. The meaning of a sentence is an emergent property that arises from the arrangement and interaction of its constituent parts, letters and words. Just as the properties of life emerge from the complex organization of biological systems, the meaning of a sentence emerges from the organization of letters and words. High-throughput technology and bioinformatics. High-throughput technology and bioinformatics are complementary because they work together to analyze and interpret large amounts of biological data. High-throughput technology generates vast amounts of data quickly and efficiently, while bioinformatics provides the computational tools and methods to analyze, integrate, and interpret this data. Bioinformatics helps to extract meaningful insights and patterns from the data, which can then be used to inform further research and experimentation. In other words, high-throughput technology provides the data, and bioinformatics provides the tools to make sense of it. 3. Regulatory mechanism in a toilet. The regulatory mechanism at work in a toilet is an example of negative feedback. When the water level in the tank reaches a certain height, the float rises and triggers the lever to shut off the water valve, preventing the tank from overflowing. This is similar to negative feedback in biological systems, where an excess of a product or substance inhibits a process or pathway. In both cases, the feedback mechanism helps to maintain homeostasis and prevent excessive accumulation of a substance, water in the toilet tank or a product in a biological pathway.